Wound healing and mitosis. Now, giving this lecture is a challenge because it may seem kind of boring. Okay, asexual, so no sex, and it's just a bunch of cells dividing, and they have different stages, they have different names. But let's look at some applications because it's kind of exciting when we think about how we cut ourselves by accident, and my gosh, that tissue is going to repair itself. Well, it's traumatic. Our epidermis is just scraped away in some sort of abrasive action, and underneath is a fragile tissue, maybe the dermis or stratum basale. If it's not covered, it's going to dry out. And that's going to slow healing because this dry, impermeable scab is not going to allow our stratum basale, our skin cells, to permeate and replicate inside this dry scab. Instead, it's a very slow healing process because these cells and the fibers have to be forced under the scab. Long, tedious journey. Mitosis is occurring, so the cells are replicating, which is what the topic of this lecture is. But what a long journey, and it takes forever that scab to heal. A more effective way is to cover the wound. And we have many different ways of covering wounds. In the hospital, it's a finesse. It's a skill that sometimes it takes years to learn how to process wound victims. Now, see, this moist environment is going to encourage the proliferation of cells. All right, so there's a good application of this mitosis. We have names for the stages just to kind of help us to learn. So interphase is when it's just preparing by duplicating its genetic material. And then after that, it's going to move on to the next stage, which is prophase. And the DNA or the chromosomes are going to thicken. They're called chromatids at this stage. And then there's these centrioles. Think of center. They're going to attach the chromosomes and to a fiber, a spindle fiber. There's the word here, highlighted. And that fiber is going to pull because we want to get these chromosomes pulled apart to the opposite ends of the cell. And that is the name of, well, metaphase is when they're on the middle, equatorial. Anaphase is when they're being pulled apart. Telophase is when they're just about pulled apart. And then anaphase, I mean, all these fancy names basically for how a cell divides. And I think I learned it when I was in school. I polish my apples twice, which I don't do. But anyway, a telophase is when there's a cytokinesis, which is a technical word meaning cell splitting. And it's splitting because now we have two. They're called identical daughter cells. And so when we have a scab, that's what we were watching. We were watching mitosis in action. And we were repairing that scab. Pretty cool. And getting back to the scab, there are stages. We have the bleeding. We know that one. But also hemostasis, because stasis means to stop. Heme means blood. And so these blood vessels are going to close down. It's called a spasm. Clots are forming. And then in a few days, we're moving on to the inflammatory stage. We've stopped the blood loss, but now we want to prevent infection. And so histamines, there's the word here, they're going to attract white blood cells to keep that wound from becoming septic. Also, the blood vessels are going to vasodilate. They're going to expand and bring lots of blood, which is what we want. Same thing when you get a cold, right? Your nose and your face gets kind of red, and we want that. The proliferative stage, we start getting some fibroblasts and collagen and start to build up that tissue. It's still fairly fragile until we finally make it to remodeling. Now, I mentioned bandages, the stratum basale. That's the bottom layer of skin that is going to make new layers. And so if you bandage a wound, you get permeation into the scab instead of underneath. right? So always put bandages. As I mentioned, bandaging is an art. You get good at it if you work with patients enough. All right, remodeling is the last stage. We start getting a tensile strength so that that skin can stretch without tearing, which is what we want, and elasticity so it stretches, scar forms. Now, suppression of collagen is something we want in certain cases because maybe we don't want a scab. Let's say sometimes someone gets a nose ring and that irritation of the metal will cause a overproduction of collagen and a keloid type of scar, a raised scar. And there's ways to use silicone coverings to suppress collagen production and keratinocyte development. It doesn't always work, but if you're in for a keloid, if it's in your family or you have a history of it, you might consider this if you ever have piercings. Dermaplaning is another application of mitosis because, okay, look at this process. And what's happening here? Collagen, elastin. Okay, movie stars pay thousands of dollars to have those proteins pumped into their skin. But scientists are finding ways that if you can trick your body into thinking it's wounded, then you can encourage the development of collagen and elastin. And one way that is done is with dermaplaning. And by dermaplaning, you take a sharp device, if I can get that to load. You get a sharp device and you scrape the top layers of skin, and here we go. And in doing so, you cause irritation. And see what she's doing there? She's encouraging the growth of collagen and elastin by wounding, basically creating tiny wounds, like microneedling also is done. Now, I don't encourage this as a beginner. Oh, she uses petroleum. I've seen it done dry. I think a good technician will do it dry. Anyway, I wouldn't do this on yourself, at least not at first, because do you see how close she was with that razor to her eye? And eyes are precious, right? We can't replace your eye. So if you're going to experiment with something like that, there's estheticians, and they are trained to do this properly. Okay, so be careful out there. Let's move on to meiosis, maybe a little more exciting because now we're dealing with sex. And during meiosis, we're going to have a reduction division because we want to make sperm and eggs. We don't want to just repair repair a scab, right? We want to produce sperm and eggs. And the way we do that is by reducing our cells to an N or haploid. So we have half the set of chromosomes. And it's really important to have this because meiosis helps us survive the hardships of life by giving us the beneficial genes of our ancestors. And that is why we don't look like our parents, right? Mom, dad, and the kids all look a little different because of different types of genetic recombination. And we're seeing that recently in Japan with, here, I'm going to talk about this in a bit. Let's stop that. Japan held the 2021 Olympics, which people were shocked, right? Because COVID. But the Japanese people, we don't know why, but they seem to have a high resistance to COVID. Now, that doesn't mean they don't get it, but it may relate to somehow beneficial genes. We just don't know. So it's kind of an exciting aspect of genetics. Is there's a lot of mystery. On to crossovers. And so how do you get all this diversity? One way is crossing over. The chromatids may break at the same place. Okay, so here are the chromatids. Remember, they're in the nucleus. And if they tear and then attach to their neighbor, we have a crossover. It has to happen with homologous or chromosomes that are the same. Okay, it doesn't happen with chromosomes that are far away. And we get that unpredictable outcome, like a brown and a blue eye. And if we take a look at this process, it makes sense because During meiosis, okay, let's take a look here. Some of these are kind of videos I dug up that other people produced I really like. Good review here. Here are those are centrioles, right? There, centriole. Chromatids. Oh, there's the spindle fibers. Yeah. Spindle fiber is going to shorten and pull. This is metaphase, right? Metaphase. Anaphase. Telophase. Cytokinesis. Man, that was a great review of just what we just talked about. So that's how it's supposed to happen, right? But if if they pull and they stick to each other, we'll just stop there. Then we have problems because then we have this crossing over, which it may not seem like that bad, but in some cases it can be fatal. Another way that genes are different is through translocation, and this is when they're not homologous. A little more severe because homologous chromosomes means they're almost the same, but if they're non-homologous, means they're from far apart, and they may code for eye color and the shape of your toe. You see that? So one example is leukemia. In the case of leukemia, okay, this is a normal type of blood. You have these white blood cells, such as monocytes, neutrophils, and you have a good distribution of these defensive cells. But in leukemia, they call them leukemia cells, but they tend to be leukocytes that they produce at such a rapid rate, they suppress the production of red blood cells, and so we can't absorb oxygen. So anemia is one symptom of leukemia. Occurs in the bone marrow, typically. Spermatogenesis, if you look at the root words here, think of the Bible, right? Genesis or beginnings. Sperm, well, the beginning of sperm. Some of these words make sense, right? Look at them. Okay, seminiferous tubules is like spaghetti. If you jam spaghetti into the testes, that's a seminiferous tubule. These are diploid when they start out, spermatogonia. And so it's kind of, think of it like a high school dance, the young cells are near the wall, they're scared. And as they move towards the center of the tubule, which is right here, we're looking at this, right? They're going to get more bold, and they're going to boogie it down on the dance floor, and that is a sperm. A sperm is in because we have meiosis. See, there's a reason we just learned about meiosis, because now we understand how we go from 2N, diploid, to haploid in, carrying half. So that way the father has half, and the mother has half. And then we get that new individual that's going to be a little distinctive, not quite like mom and dad. Oogenesis, the beginning of the egg. A primary follicle, which is really tiny. And this is why a woman wants to have children, hopefully before she's 45, because her whole life, those primary follicles have been exposed to all kinds of radiating factors, irradiating factors. I'll talk about it another time. Oocyte is the egg, and it's surrounded by a pellucida, which helps nourish it. Now, estrogen is going to cause this cycling to occur so that this primary becomes a secondary, and it's going to mature into a graphene. Here's a graphene right here. And we're going to see a better look at a graphene. I just want you to see where we're at, right? We're in the ovaries of the female reproductive system. All right. Let's take a look at the graphene. So here's another view of it. Primordium to graphene. Graphene contains the mature egg, which is going to be released, and it's a really specific process, right? It's going to go from the graphene out of the ovary, and we call this ovulation. And then here it goes. Okay, I really like this video. It summarizes what I'm just covering. So here's the egg. I'm sorry, here's the ovary. Here's the ovary. There's the fimbriae. Okay, those are finger-like projections of the uterine tube or fallopian tube. Now, estrogen is going to cause the thickening of the endometrium. There's the word spelled right there. So the egg can implant. All right. And here comes the egg. It's leaving the ovary. So here's the ovary. It's leaving. The fimbriae are going to sweep it. Isn't that cool?